Okay, so welcome everyone to this innovation workshop on the final day of Genesis 2022. So our workshop today is in collaboration with Kobe Biomedical Innovation Center, or Cluster, sorry, excuse me, who are going to present a startup pitch comprised of 4 cutting-edge companies with exciting technologies who are looking to partner globally. So just admitting participant, thank you. So just some housekeeping. As with the previous workshops, the chat box will be enabled, so please feel free to use that to submit any questions. The format of the session is going to be 4 videos, and we're very lucky to have the contributors of those videos joining the workshop, so they will be available to answer your questions, and we'll take questions after each of the videos is played. So with that, I'd just like to hand the floor now to Keiko Kobayashi, who has very kindly agreed to moderate today's session, and will be introducing each of our speakers, and will also be saying a little bit more about the Kobe Biomedical Innovation Cluster. So Keiko, I'm going to hand to yourself, and I really look forward to hearing the presentations. Thank you very much.
Yeah, thank you very much, Alicia. Hello, everyone. My name is Keiko Kobayashi, a member of Foundation for Biomedical Research and Innovation at Kobe, FBRI. So thank you very much for your participation. Today, I would like to introduce about Kobe Biomedical Innovation Cluster and 4 startups who are seeking to find business partners globally. So let me share my screen. Okay, so let me introduce about the Kobe Medical Innovation Cluster. So KBIC is the largest biomedical hub since 1998, and it is aiming to attract healthcare-related industries. And currently, there are around 360 companies and organizations related to life science having their R&D activities. So this pie chart shows the breakdown of KBIC members. Medical device related and then pharmaceutical related companies accounts half of them. And there are some regenerative medicine and healthcare related companies, including some of the foreign affiliated companies such as Bayer and Boehringer Ingelheim. And together we promote the open innovation activities. And there are also some university campuses and including some R&D facilities like RIKEM. And the graph on the right shows the number of KBIC members. As you can see, it has steadily increased, and as the number grows, it is giving the positive impact to Kobe City's economy. And this is the overview of KBIC. It can be divided into 3 subclusters. Medical cluster houses 8 highly specialized hospitals, and the middle one is a biocluster, which is housed for well-known research institutes such as RIKEM. And finally, the simulation cluster houses world's fastest supercomputer, Fugaku. So in this environment, KBIC member companies are conducting their research and business development activities. Foundation for Biomedical Research and Innovation at Kobe, FBRI, is a main supporting organization of KBIC. The president of FBRI is Dr. Tasuku Honjo, who won Nobel Prize in Medicine or Physiology in year 2018. And under FBRI, there are 4 centers. There are Center for Basic Research, Translational Research, Cell Therapy and Cell Manufacturing. And the last one is the CCD, is a center for coordinating the different players in KBIC. So, and our focuses are pharmaceuticals, medical devices, regenerative medicine, and healthcare. And to support activities within KBIC, dedicated coordinators with a medical background are supporting each of the member companies. The kind of support FBRI provides includes regulatory affairs related advice, recruitment, and human resource development, providing business matching or networking opportunities, and so on. And one of the supporting functions of FPRI is to coordinate joint activities with hospitals in KBIC. As I previously mentioned, there are 8 hospitals in KBIC, so we, FPRI, played a role to connect companies with hospitals. So for example, if a company wants to collaborate with one of the hospitals, FPRI identify the right partners so that they can start joint development and joint activities. And since a couple of years ago, we are focusing on supporting startups, especially at their early stage, because startups in that stage needs much support. We provide a kind of tailor-made support for startups, such as providing the advisories, various training program, office spaces with discount, useful subsidies, and so on. So why startups choose KBIC? As I explained in my previous slides, there are all the elements in KBIC that startups need to grow so that they can accelerate their business development. And currently, among those 360 companies, there are about 70 plus startups in KBIC, and we expect these numbers to grow. And today, I'm very happy to have this opportunity to introduce 4 startups who are operating their business within this COVID startup ecosystem. They are CarbGen, Progenicide Japan, BC Cell Therapy, and EMO. So let's move on to their pitch session. During the video session, if you have any questions, please put your comments on the chat box. Okay, so is Mr. Yang here? He is not here yet. Okay, so Dr. Sugaya, can you go? Can I go your presentation first?
Sure, of course.
Okay, thank you. So let's start with Progenicide, please.
Hello, I'm Kim Sugaya, CEO of Progenicide Japan. Progenicide Japan is the spin-off company from the University of Central Florida, established in the middle of the Kobe Biomedical Innovation Cluster in January 2020. It aims to put the innovative technologies developed at the College of Medicine, UCF, to the market. UCF has been closely working with NASA to develop advanced engineering, such as nanotechnology, optics, computer science, electroengineering. Today, I'd like to introduce rapid detection of multiple pathogens in the air. Biosensor-based immediate detection of the pathogens with the comparable sensitivity to conventional PCR is the technology we developed. Before that, I'd like to talk about what we have done. When the patient comes into the clinic with the cold symptoms, such as fever, cough, sore throat, we don't know which pathogen causing this. Then we have to send out the subject sample for the PCR test. But the current PCR test has lots of issues, problems. It takes time to transport the samples to the special facility. And also it costs because such a special equipment, facility, and the skilled technicians are needed. And these times and costs increase by the number of the possible pathogen involved. You have to test them all, otherwise you never know. But we gave the answer to this. We developed the handheld real-time PCR machine which does not require isolation or purification of nucleotide from the sample. Using this, anybody can do the PCR anywhere to test, and then such a result can be found in the 20-30 minutes. That's the totally different from the current PCR testing. This technology can amplify the sample without the nucleotide isolation, like this. Very specific amplification we can get. But without our technology, using the conventional PCR, put the sample just into the PCR tube without isolation of the nucleotide. It gives you the non-specific amplification like this. That way we cannot diagnose, but with our technology, it's clearly diagnosed. But we have a next generation technology developed, which doesn't require the PCR. That means you don't have to wait 20 minutes, 30 minutes to find the results. In few seconds, you can see the results. And then this blue, red, those are just a basic sensor color. You don't see any changes in the wavelengths. But with the media, let me explain how we get this sample. We culture the normal human lung fibroblast and then infect with the human coronavirus. And then we just take out the 5 microliter of the media, spent media from the culture and put on the biosensor. That's what we did. So again, this gray is just a media without a virus, just a cell culture media. And then this orange or yellow, you can see tremendous difference. And this is the triplicate the results. You can see such a difference here. Okay, and then that one from the culture media, just 5 microliter. We don't need much. So, and then when we check this media with the conventional PCR, it takes more than 30 cycles to detect such a virus. Very low viral particle in this sample. So this means we can detect the virus in seconds without PCR amplification, with very high sensitivity. Using this, we developed like this one. We're gonna develop the machine which has an air sample here, which capture the pathogens with the high efficiency, and then bring those sample to the biosensor, which I explained right now. And then less than a minute, you can see the results. And then if the pathogen exists in a sample, it will beep or make a light, warning light. And then we know we are in a danger. We have such a virus or any pathogens in the air. We can test this one against the multiple pathogen at once. This is the beauty of this machine. So now we are looking for the partner to develop this machine with us. This is the contact information. And then, of course, you can visit our website, which is here. Thank you very much for your attention.
Okay, so Dr. Sugaya, can I ask
Thank you.
a few? Yes, thank
Sure.
you very much. Can I ask? So, okay, as for your device to detect the pathogen in the air, is the prototype completed?
Not quite yet. Although we have a basic technology already developed, but we need to put two together and put in a compact body. That's part still we're working on it. But almost there.
Okay, thank you. And the next question, how many types or kinds of pathogen can you detect at the same time? And how large, how big the space can it detect the pathogen with your device?
I see. All right, so then the pathogen-wise, theoretically, we can put the thousands, but that makes that requires the high-resolution pickup camera or something like that. But easily we can put the 100 or something pathogen at once. And then talking about the space, covering the space. This machine we're developing the tiny like this size. So then it covers maybe like this room. So not that huge area. But if you can put this device, several of those, then you can cover quite a wide area. And then maybe the next generation we can, if it has so much demand, maybe we can develop the huge one and then cover the like in the airport or something like that. So, but anyway,
wow,
that's a dream.
Good. Okay, then okay, my next question is, how much do you think the price would be for the product?
We didn't want to say that at this point, but I mean, it's, let's say, we're gonna maybe have like this PCR machine 10 times less than regular PCR machine. And then that gives you the kind of idea. And then maybe the affordable for the individuals at the home. That's kind of pricing. That's what we are expecting.
Okay, thank you very much. So yes, if you have any comments or for the audience, what kind of partner you are seeking, please speak to us.
Oh, yeah. From my side, I like to see some of the collaborator to make this happen, especially we're a startup company. And then we have a technology, we have a dream, we have the idea, but to make that happen, especially put into the mass production, that's the part a lot of still hurdle. So that's the part. If we can have a partner to make that happen, that'd be great. And then we love to put these things as soon as possible. Yeah, as you can imagine. And then this is not just for the COVID-19. Even you can use for this one influenza or any of the virus, any of the pathogen floating in the air. So that's the beauty of this machine. So please contact us.
Okay, great. Thank you. Thank you
Hello,
very much.
sorry, it's Alicia here from One Nucleus. Please, may I ask a question? In terms of once you have the initial prototype developed, the concept we've just seen, where do you expect that to be applied? Is that something that would be rolled out in hospital or clinical settings, or is it something maybe more vulnerable people of the population would be given access to? Where do you see it sort of being applied initially?
Yeah, for the prototype, maybe we love to use the hospital environment because we have a patient. We know that's the environment, already contaminated or not. So then that's a good testing place. But we are aiming even for just like restaurants or even the trains, because this is a portable one. Even you can bring into the airplane and then just check in your environment right there. So that's the kind of things we are thinking.
Thank you very much. Thanks.
Thank you very much, Dr. Sugaya. So I hope somebody would show interest.
Yeah, I'd love to
Yes.
hear something. All right.
Yes. Great. Thank you for your time today.
Thank
you
you.
very much. Thank you. So, okay, let me move on to the next startup. Next startup is VC cell therapy. Let me play the video. Hold on, please.
Can you hear me?
I will play. Actually, I will play the
Our
video.
company is called VC Cell Therapy, and we are providing transformative regenerative medicine for ophthalmology care. My name is Somikyo Tani, and I am a director at the VCCT. What we would like to solve is retinal degeneration. Once diagnosed, patients go through gradual loss of vision and become unable to read or recognize faces of loved ones. Because the disease is progressive, patients upon diagnosis often become devastated in their future and isolated in society. Our founder, Masayo Takahashi, has been consulting these patients over 30 years as an ophthalmologist, and she founded this company to address this very issue. Retinal degeneration is caused by destruction of retina, which is a thin film situated at the back of the eye. Especially, loss of two cell types are responsible and cause blindness: photoreceptor cells that are light sensing, and RPE cells, which provide nutrients to photoreceptor cells. Once they are broken down, they cannot be regenerated naturally, and current treatments often only slows down the disease progression without curing. Retinal degeneration comes in many types. Depending on etiology and how the disease progresses, many different names are given to the disease. Retinitis pigmentosa is genetic disease affecting 1.5 million patients worldwide, while age-related macular degeneration is a disorder affecting 200 million patients worldwide. Currently, there is only one medication really in the market targeting a subtype of AMD. Many are in development, but only the cell therapy is really curative as opposed to preventive measures. Our solution is conceptually simple. We make cells in the lab that are lost in the degeneration. We use allogeneic off-the-shelf and differentiate into RPE or photoreceptor cells using own protocol. Importantly, we then process these tissues as such that they can be easily transplanted into subretinal space using routine clinical procedures. We have 4 pipelines of regenerative medicine internally. First 2 are only for Japan, bringing RPE therapy to market in an expedited pathway and generating cash early from 2023 or 24. On contrary, we are bringing more universal RPE and photoreceptor cells to the global market. They are currently in preclinical development. We have accumulated clinical evidence for safety and efficacy. As you can see, when we transplanted RPE cells in patients, they could survive within lesion for over 4 years. And importantly, RPE cells could support the nearby photoreceptor cells healthy. For this patient, we could observe that the visual acuity stabilized after transplantation despite progressive nature of this disease. This is even after patients stopped previous medication. Furthermore, we could see through the survey that the patient's quality of life improved 4 weeks post-transplantation. We have preclinical evidence for our photoreceptor asset. It survives over 2 years without much immune rejection or complications in monkeys and integrates nicely within host tissue. Furthermore, we see in rats that these transplanted cells become responsive to light and form electrical connections to downstream neural network. So what is so special about our company? It is unique in our products being most applicable and safe compared to other products in the market. Retinal degeneration encompasses different manifestations of disease, and only 30% of patients have RPE degenerated, but photoreceptors somewhat intact. However, the majority, 60% of patients, have both RPE and photoreceptor cells destroyed, and this is where only we can help. Through our clinical experiences, we realized that the drug formulation is the key to efficacy and safety. We see others use cell sheet or cell suspension formulation, either which we tried but found major drawbacks either in lesion coverage or surgical hurdles. Our proprietary cell strips, however, solve both issues and can provide best outcome for patients. We experienced also that the variation in quality of cell therapy product is problematic and caused by human techniques. Hence, we copied our best technicians' handworks to a humanoid robot, Maholo. Now, manufacturing is very robust and it is very easy for the tech transfer. We are envisioning two different business models. One is where we directly provide cell products to our partner hospitals in Japan. We have capacity ready to treat about 120 patients annually, and this could make up to 15 million USD per year. Another is through partnering. We are talking to two pharmacos, one of which is our investor. By bringing the products through phase 1/2, we are aiming to license out the assets and generate royalty revenue. Our product is also well prepared to overcome future regulatory hurdles. We are adapting clinical grade materials and already in negotiation with manufacturers for future tech transfer. We use simple devices for surgery and routinely administered medications only. We are also the experts of trial design, stemming from years of accumulated know-how from Japan. Last of all, we have a great team. Masayo, our founder of our company, is a world-renowned KOL and ophthalmologist, and we have industry, business, and finance experts reinforcing the team. Also, our biggest asset is a clinical team led by Tadao. More than 10 clinicians who are the expert on the cell therapy are advising us on development. Thank you very much for your attention. We are always looking for potential investors and partners for development, so please feel free to reach out with any inquiries. Thank you. I'll come.
So it's your turn, Mr. Otani. Can I ask a few questions to you?
Of course.
Okay, thank you. So I understand that you're planning to proceed your clinical development in Japan first, but is there any possibility for you to conduct clinical trials in Europe by yourself? And in that case, are you thinking to out-license to overseas pharma companies?
Yeah, the short answer is yes. So we have a great capability here in Japan where we will be able to conduct clinical research and trials here, and that will be our pioneering program. But we have two assets that are specifically targeted for the global audience, and that we would not be able to perform by ourselves throughout in Europe and US. Hence that we would be looking for a development partners in both regions.
Okay, great, thank you. So next question, as for the manufacturing of cell strip, even if you conduct clinical trial overseas, do you plan to manufacture in Japan and ship out, or do you plan to collaborate with companies overseas and manufacture locally?
Yeah, thank you. It can go either way, and it depends on it all depends on the on our partners and then financial model. So one possibility is that because we are making, as you saw in robots, that the tech transfer will be quite easy. So we will basically, for example, work with a partner in Europe, and then they will be able to manufacture. That's one possibility. And another is we are investing heavily in this frozen format, so that it is possible that we can actually produce it in Japan and then ship it. But it's all because of the robustness of the manufacturing protocol now being automated. We'll be able to take either way, and depends on the financial benefit.
Okay, understand. Thank you. And then my last question is, and I understand that in the field of ophthalmology, the clinical development of gene therapy is quite advanced. But what do you think is the advantage of regenerative medicine versus gene therapy?
Yeah, thank you. So obviously both have advantage in some areas and others. And if you think about the retinal degeneration, it's a heterogeneous disease. And then about the part of that is caused by a gene called retinitis pigmentosa. and that affects about 1.5 million people globally. And that obviously will be best targeted by gene therapy, perhaps. On the other hand, some of the disease, for example, age-related macular degeneration, they are not necessarily of genetic origin. Gene plays as a genotype is a contributing factor, but they're not the determinant. So in that case, that gene therapy may not necessarily work. So we will also go in as using a cell therapy to treat these patients.
Okay, thank you. So yeah, if you have any comments or anything that you would like to talk, please go ahead.
Thank you very much. It's just Alicia from Nicholas again. I just wanted to say, I think this is a really important focus area, given that conditions such as the ones you talked about could be related with sort of aging populations. And so as we move forward and, as populations do age in different around the world, it's now becoming more of an issue. I think it's definitely one of the focus areas. And I was really interested actually to see the slide on automation. So perhaps that is the key when we're looking at these therapies that are difficult or have challenges in scaling up in manufacture. Of course, there'll always be a human element needed, but I think that was a really pertinent point as well around how automation is enabling us to achieve that robustness of manufacture as well. So yeah, really, really interesting. Thank you, Mr. Otani.
Thank you very much.
Thank you. So if you have any comment, it's okay. Okay, all right. Thank you very much, Mr. Otani. Thank you. So let me move on to the next startups. It's IMO. So let me play the video. Hold on, please.
Hello everyone. Thank you for having me here. My name is Daiki Takegawa. I am COO of IMO. We date anxiety disorder from this world. As you know, many people with anxiety disorder do not receive appropriate treatment. Treatment guidelines recommend either pharmacotherapy or cognitive behavioral therapy. It is called CBT for short. But pharmacotherapy is often used regardless of severity in Japan. Here is a summary of each treatment. Pharmacotherapy has significant side effects. CBT is preferred for mild to moderate disease. Why is appropriate treatment not currently available? There are three main problems. First, different programs for different disorders. A number of CBT-based programs are being developed and the program differ for each disorder. Second, only spend 3 minutes of each patient. Doctors have limited time to spend with each patient, making it difficult to provide CBT. Third, CBT provided by counselors are not covered by insurance. In Japan, CBT provided by counselors is not covered by health insurance. So patients have to pay 100% of the bill. Here is our product IMO, a CBT-based mobile application for the treatment of anxiety disorders. Our solution is to provide disease-specific CBT-based mobile apps with insurance coverage. In this app, patients receive a program based on CBT by interacting with an AI chatbot. So, there is no need for human intervention. We are developing an app to treat two major anxiety disorders. Both are very important diseases. This graph shows difference in effectiveness of different delivery formats of CBT. Results of the research suggest that self-help and individual psychotherapy in person are both viable treatment options for depression. DTX products for mental health are rapidly increasing. but we are aiming to be the best in the world at apps for treatment anxiety disorder. Our biggest strengths are high retention rates, collaboration with key opinion leaders, and a well-developed CBT-based program. The completion rate of the e-mod programs is 92%. These are our research results. Significant results in social anxiety disorder and mental health status. This is a preliminary study on a small scale, but we plan to start our clinical study next year. From Japan to the world, we are looking for partners to develop an app for the treatment of anxiety disorders. Thank you so much.
Thank you very much. So, Ms. Hata and Mr. Takegawa, let me ask a few questions to you. I understand that you will be developing your apps for obsessive compulsive disorder and social anxiety disorder as the target diseases, but what is your expectation in terms of the market size of these diseases?
Thank you for the question. Today, regarding the proposed questions from Ms. Kobayashi, as an interpreter, I would like to share the answers I received from Mr. Takegawa beforehand. The answer would be the estimated number of patients with OCD is about 2% of the population right now. And the estimated number of patients with SAD will be about 12% of the population. The medical cost of mental disorders in Japan amounts to 1.9 trillion yen, and the estimated market size of anxiety disorders in Japan is about 300 billion yen. So we believe that the global market for anxiety disorders is expected to be more than 10 times that amount.
Okay, thank you very much for your information. All right, then let me ask you the next question. So please tell us about the details of a clinical trial that you are planning to conduct next year for obsessive compulsive disorder and social anxiety disorder.
Thank you for the question. First of all, the clinical trial we are planning for next year will be a single-armed intervention for obsessive compulsive disorder patients. Then, based on the result of that study, we are planning to conduct a non-inferiority trial with CBT, cognitive behavioral therapy, in person.
Okay, great. So my next question, would you tell us about your plans for your product development in Europe?
Yes, we have a plan to start a clinical study also in Europe in parallel with the clinical study in Japan, immediately after the specific clinical study next year. So this will, how it will go will depend on the results of the clinical trial in Japan conducted next year, but we are planning a trial in Europe.
Okay, and I also want to know about your business model of your product development in Europe.
Thank you so much. In Europe, the insurance system is similar to the Japanese insurance system. So we are considering that providing this application in the form of a prescription at a clinic after it is approved as a medical device.
Okay, noted. So my last question is, what kind of business partner in specific are you looking for in Europe?
Thank you so much. We are still continuing to develop the product in-house, but we are also looking forward to coordinating a local clinical research and the sales partnerships in Europe. So we are waiting for any partners to be to work with us.
Great. Good luck with your finding your partners. Thank you
Thank
very
you so
much for your
much.
time. Thank you.
Thank you so much.
Okay, so let me move on to our last startup, which is CarbGen. Please hold on, please.
Hello, everyone. I'm Luda. I'm honored to be here today. I work for the company called CarbGen. It's a Japanese healthcare startup focused on digital and other solutions in antimicrobial resistance and the infection disease area. Today, I would like to present our technology in the following session. So this is today's agenda. After the brief introduction of our company, I will present our technologies and products. In the end, I will talk about the partnerships and opportunities we are looking for. This is the snapshot of our company. We just funded last year. Our business mainly focuses on products in infection disease. Our seed technology is inspired by the clinical needs of National Center for Global Health and Medicine in Japan. The institution is similar to American CDC. These are the facilities and the institutions we have been working with. Most of our development projects are supported by government grants right now. This is a profile of our management team. The members have a proven track record in business, healthcare, and IT industries. Then I would like to talk about our products. The background of our development is the problem of AMR, or antimicrobial resistance. It's a global threat. Nearly 10 million people may die directly by AMR in 2050 if we don't take a change right now. The number is more than people died by cancer. These are the potential actions against the AMR, and all focus is to promote proper prescription of antibiotics by using the digital technologies. In general, the de-escalation process contributes to the proper use of antibiotics. In that case, doctors will change prescription, the broader spectrum antibiotics to narrower spectrum antibiotics, but there is no fast and accurate way to identify the causative bacteria, especially in the early stage. So the current process is subjective, time-consuming, and expensive. So the Grimston, let me talk about the product concept. The Grimston and macroscope observation can identify the causative bacteria by methodology, but the accuracy is not so high. Then we realized if we can train the AI with a lot of Grimston images, we can improve the accuracy issue. And also the application can link to the antibiogram, which will also recommend the antibiotics. So after user interviews with doctors, it suggests that it's better to have additional function to let the doctors to check with other more experienced ones in infection disease. So this leads to the development of our telediagnosis system, CarbConnect. So another product concept is POX. So with further discussion with doctors, we found that Grimston itself could be a bottleneck to introduce our AI system into the medical facilities. Although Grimston is a well-known diagnostic method, it's not usually conducted in daily operation. In the left side, you can see Grimston is with a lot of process. Physicians may fail to make a valid Grimston slides, but with the help of a point-of-care device, the Grimston can be done easily. So here is a value proposition. The POCUS will enable automation gram stain precise, and the BTA can provide a high accuracy and rapid diagnosis in bacterial identification. Besides, it will also provide evidence-based antibiotics suggestion. The CAP Connect is capable of telediagnosis and can also provide a second opinion for the doctors. And all target customers are from medical facilities and clinical labs. So here is a workflow of how to use POCUS and the BTA. After the spread of the sample to the plate, the gram stain can be automatically processed by the POCUS by uploading the images from via a smartphone to the AI deployed in the cloud or local server. The application can give the name of the bacteria. With the reference of the antibiogram, the app can also make a suggestion on the antibiotics. So let's have a look of the demo video. Hope it can give you a better understanding of our products. So next is a slide about the comparison of diagnosis methods. This one shows you our application and the difference between our application, our products and other diagnosis methods. As you can see, with the help of the AI and automation technology, our products can provide more accurate, if low cost, speed, and convenient solutions in bacterial infections disease diagnosis. So here is the pipeline and the development timeline. So in Japan, we are preparing the regulatory approval and will submit the application next year. Meanwhile, we start to work with institutions overseas to develop the AI system. Beyond our collaboration in overseas, we continue to work closely with potential customers. So far, we have interviewed or surveyed over 50 key opinion leaders in infection disease, and almost half of them are products. Lastly, I would like to talk about the partnerships and opportunities. So currently, our AI is only trained with the urine and blood samples. We want to work with partners to expand other type of samples. And meanwhile, we are looking for partners in overseas to help us to achieve the data acquisition. In addition, we are also hoping we can have a partner in manufacturing. Thank you for listening. Please contact me if you have any questions or if you're interested in our technology. Thank you.
Okay, so everybody, Corruption is not here, unfortunately, to answer your Q&A. So For the audience, if you have any questions to BC cell therapy, progeny site, and IMO, please, you can speak out or you can put the question in the chat box.
Apologies, I'm muted. Just to add as well, you can use the conference app to continue discussions with Kaiko, who can also feed back to each of the speakers. I just want to take the opportunity to say thank you very much to Kaiko for running this session and for making the introductions to the speakers. Dr. Sugaya, Mr. Otani, Mr. Takigawa, and Mr. Hata, and then Mr. Yang as well for his presentation. It was really, really interesting. Lots of exciting technologies and opportunities to expand the capabilities of the technology. So I guess the point of the session is to create those connections to hopefully expand the opportunities here. So please, if you have any questions at all or any comments, don't be shy. Feel free to say something. And the session has been recorded as well, so you can always catch up on any of the content if you would like to.
Nobody has
Quiet,
questions.
yeah. I mean,
So quiet.
yes.
Yeah.
From the perspective of those who presented, in terms of when you're looking to partner, what kind of is there certain criteria you're looking in your collaborators? Mr. Sugaya, you're on the screen. So did you want to say anything more? Or sorry, excuse me, Dr. Sugaya, about the qualities or the criteria you're looking for in your partner or the specific area? Because you said you had other opportunities as well. So there's some time if you wanted to talk about that as well.
Yeah, actually, right now we're seeking the mass production of this sensor. The sensor, structure itself already established, but it requires the nanotechnology etching to make it, like UFA for the CPU. So then those things, those technologies we are looking for. And then in Japan, already we are identifying those companies, but we want to distribute this to all over the world. So then that means we need more of those companies. And also of course, always the funding is welcome. So then the angel investors or even the VC welcome. So that's the thing. Just I want to say that I developed these things because I hate the coronavirus, prevent us to meet face to face. So then that's the major things. That's why I developed this PCR machine as well. And then now we have a technology to kill the coronavirus using the exosome delivery of nucleotide. So those things we, put together. I like to go back to Japan. I used to go to Japan twice or three times per month, but now no way. So that's the things we want to solve the problem. And then I saw the very nice technology about the cell therapy as well. That's my major area, actually. We have been working on the stem cells, and then and that's a very good technology, amazing. So we like to see those things more developed.
That's upcoming areas, indeed, modalities that I'm sure there'll be more research into. That's really interesting. Thank you very much, Dr. Sugaya. Kaiko, would you like to say any final words around the innovation cluster in Kobe. So for any of the attendees, if you had to say something to promote that center of excellence, what would you say?
Yes, so thank you very much again for this opportunity. And for startups that we presented today, I'm sure that they are very much with the cutting edge technology, and many people would show interest. So please make this video as an archive and then spread to your network. And some people could not join today, but I'm sure that they will be interested.
Excellent. Thank you very
Yes.
much. And thank you to everyone who joined the session today. So I hope you've had an insightful time listening. And of course, if you attended previous sessions, I hope you've enjoyed those as well, and the in-person day. Please continue to use the conference app to connect and to send messages and to learn and to meet new people. That's why we have it here, and that's why it's going to be continued. You can continue using the app for the next 4 weeks as well. So please make best use of that, and I wish you happy partnering in that respect. So thank you very much, everyone. I'm going to bring the session to a close now. Thank you all.
Thank you very much.